Oh, thank you for inviting me for this conference. And this is a slightly different topic. Um, I'm from New Zealand. Jingy will be sharing the talk in the middle. And let's see whether we can actually complete within 20 minutes. So our emphasis is actually on modeling the Alzheimer's disease, especially the neuronal dysfunctions. And also we are trying to understand the disease in terms of biophysics. So this has some bearing on developing mathematical models and then testing them. And most of the work we are doing is actually based on experimental results. So that's we gather quite a bit of experimental results to do our work. So the talk, we'll start with just introducing to Alzheimer's disease very briefly. And then the reason I want to introduce that is from a modeler's point of view. I'm looking at especially a neuron as a system, basically a system with boundaries and molecules interacting. And then the system has subsystems as well. So, and one of the subsystems I just want to touch on is AIS region, which has a lot of work done where the action potential starts, and that has some bearing on Alzheimer's disease. And then Jingy will start talking about the modeling and investigation of NMDA-mediated calcium signaling. And this is PhD work in Lincoln. So in terms of Alzheimer's disease, you know that these statistics, our keynote speaker talked about this. And this, I mean, we are going through a COVID pandemic, but I think in time to come, most of the countries will face huge costs. So this is going to be more and more important. And then you know that the basically accumulation of proteins, plaques, and tangles, and also the brain shrinkage, and we have 3 different stages of Alzheimer based on how it progressed. But prior to this, few years prior to this, the onset is actually starting. So one of the things we are trying to do in my computation lab is trying to figure out when onset starts and where, things like that. There are a lot going on. Just a brief summary of this work is presented here. And what we are looking at is basically these neurons as single cells. We have been working on single cell dynamics for the last 20 years. And then neuron is a special cell which has action potential, electrical signals going through in one direction. So there are stimuli coming, and then there will be gradients, and then go into the action potential. So in terms of, if you look at this interaction between 2 cells, is through action, and then electrical signal is passing in and going into the synapses and go to the next cell. So our focus is actually a single cell, and then also this process of moving. And this one is actually initiated with the collaboration of Peking University, China, in their brain institute. And Professor Zhang Zhang's lab actually worked on AIS region. And we got involved. Essentially, I got the idea to model this. This region here is very important in terms of generating the action potential. This is where the action potential starts and then move in unidirectional manner. And there's a lot of work done on this, and we actually develop a model and publish some of the activities within that. This region is about 40 micrometers at length and 1.5 micrometers of diameter. And it's a fairly small mesoscopic scale, so it has its own challenges in mathematics of modeling this. And it also have a special purpose of actually have a unidirectional action potential going through this. So if you look at the membrane of this, the way I'm not going to explain how we model this in detail, but we took this particular anchoring G as a molecule, and it actually binds the sodium and potassium channels, and those dynamics, more sodium channels get bound to the membrane, then you have a high probability of generating action potentials and going through this. So there's quite a bit of dynamics here, which we model through mathematical and computation models, and we publish this in a paper. And this is how, if you want to look at the interactions within biomolecular interaction, this is a diagram which shows that it's quite a bit of complexity in this, and because of that, we need fairly sophisticated mathematical and computational framework to model these. And we have done that in Lincoln and publish these things in our papers. And this is another diagram to show that how within the axon AIS region, the activities are related to each other. So this is a fairly systemic functions. It's a systemic way of looking, developing models using these kind of interactions. And in terms of, I think AIS region has a fairly important role in Alzheimer's disease brains. So this has been established experimentally in Peking University, and we have shown that in our modeling work, it is the case. And if you look at the role of AIS region, there's quite a bit of work, and now we know, especially we look at this link here, the link between AIS region dysfunctions and Alzheimer's, and especially related to anchor G downregulations will trigger a few things. So we have actually worked on a specific model related to that. And in terms of the modeling, we have fairly sophisticated frameworks and also methodologies to model these interactions. And we use different approaches in mathematical and computational modeling, as you see. And we also take physiology as well as whatever important essential interactions are taken into account. So this has, and some platforms like we sell are quite useful in this regard. And I'm going to pass this to Jingy to start her presentation within the next few minutes. Jingy.
Okay, thanks, Don. Now I'm going to share my screen with everyone. Can you see it?
Yes.
Yeah,
Okay. Hello, everyone. My name is Jingyi Liang. Now I'm going to present a model as an example to show how the computational model approach can help scientists understand the dysregulation of AD. We have used this model to investigate the NMDAR-mediated calcium signaling in AD. First, I'd like to give you some background information of the model. So the NMDAR-mediated calcium signaling happens at the synapse, or more precisely within the postsynaptic span. This is the typical structure of chemical synapse. When action potential reaches to the presynaptic terminal, it induces the release of neurotransmitter into the synaptic cleft, and the transmitter can diffuse and bind to the glutamate receptors at different locations. One type of the receptor is called NMDAR. It has 4 subunits, most comprised of two NR1 and 2 NR2. And for the NR2, there are NR2A and NR2B subtypes, lead to different affinity for glutamate by NMDAR. And the models I'm going to present today also focus on the differences between NR2A and NR2B types of NMDAR. NMDAR have been found at many different locations, and it is the major type of calcium channel contributing to the postsynaptic calcium response. In Alzheimer's disease, amyloid beta is the main component of the amyloid plaques. Amyloid beta has the ability to self-associate and exists in many different forms. Experimental evidences have suggested that amyloid beta oligomers are the most toxic form. They can also interact with multiple key proteins in synaptic transmission, and calcium signaling pathways ultimately result in nerve damage. Amyloid beta oligomer is proposed to cause neuronal toxicity through different pathways. The calcium hypothesis is one important hypothesis that extends from the amyloid hypothesis to further explain the alteration resulting from amyloid pathology. Calcium dysregulation have been observed in brains of AD patients before the presence of the clinical symptoms of AD. The deregulation of calcium signaling is suggested to account for the decline in cognitive functions in the early stage of the AD, as well as the neuronal loss in the late stage of the AD. Amyloid oligomers also interacted with multiple key proteins in glutamate transmission pathways. Here are disturbances here are disturbances by the amyloid beta in AD, referring to the arrow labels over there. One is amyloid beta inhibits glutamate clearance by the transporters. 2 and 3, these are the non-synaptic glutamate release. And 4, this is the internalization of the surface receptors at the synaptic site. 1 and 3 will lead to an increase in the extracellular glutamate concentration and ultimately may lead to the overactivation of the glutamate receptors. And 4 will lead to a decrease in the functional synaptic receptors and may depress the synaptic activity. So based on those observations, we are going to use a computational model to answer these 2 research questions. First one, how do amyloid beta disturbances under AD condition can affect the synaptic transmission and NMDAR-mediated calcium signaling? 2, how do synaptic NMDAR alterations disrupt the downstream event? Here is a conceptual framework of our model. It includes 3 submodels. First, it is a two-dimensional glutamate diffusion model. These arrows denote the diffusion of the direction of the glutamate, and these 3 labeled regions represent different locations of the receptors. So we simulate the glutamate concentration at these 3 different locations and send the concentration to our model 2, which is simulate the receptor activity. So this is the second submodel. It simulates the state transition of the NMDAR mediated by the glutamate. So for NR2A and NR2B types of NMDAR, they share the same activation schemes, but with different reaction rates, rate constants. This means we can investigate the effects of the different types of NMDAR. The full opening of the receptor depends on both glutamate concentration as well as the membrane voltage. So we also have a membrane voltage model built in. This is the last submodel. It is a compartment model of the calcium dynamic. It includes 4 compartment models, which represent a pyramidal dendritic span, and it is adjacent dendritic shaft. So once we got the model, we estimated the unknown parameter and validate the model against the experimental data. Then we identify the key parameters which related to the alterations by amyloid beta. Then we can start our computational experiments to answer these 2 questions. For the first one, the alteration by amyloid beta are going to be investigated at the availability of the glutamate and the availability of the receptor. To mimic these alterations, we manipulating the key parameters related to those alterations and run the simulation separately. These are the results of our simulation. When we simulate the disturbance in glutamate availability, we can see different alterations will affect the different receptors, and the elevation of the resting glutamate level, it may not induce the receptor opening, but it will reduce the receptor sensitivity long-termly. And also, when we simulate the disturbance in NMDAR, reduction in the NR2A numbers dramatically reduce the postsynaptic calcium response, but reduction in NR2B type NMDAR shows minor impact on it. It suggests the proposed neuroprotective function of the NR2B type NMDAR internalization may be very limited. We also have done a global sensitivity on the selected factors related to the alterations, and the heat map shows if we look the heat map vertical from a vertical direction, that means from the point of view of the parameter contribution, all outputs, but not the calcium number entering through the synaptic NR2B type NMDAR are very sensitive to the amount of the presynaptic release of the glutamate. And if we look at the result the other way, we can find that the calcium transient in the span hand is the most sensitive to synaptic NR2A type NMDAR. Then to answer the second question, we added another model to our current ones, which represent this downstream event. So in this simulation, we want to see how different types of the NMDAR will affect the calcium influx as well as the downstream event, we found NR2A type of NMDR is mainly contributed to the calcium influx, while the other one is mostly contributed to the downstream event. Also, we have changed the ratio between these two types of receptor. And simulation with different ratios suggests that although a high number of the NR2A types of MDR is very important, we still need some numbers of the NR2B types of receptor to finish the downstream event. So these are the results of our simulation. I'm going to skip the future direction and hand the presentation back to Don.
Yeah, I think this is just the last slide. If you want to understand what we have done, there's a book we wrote, came out this year, and also we have few papers online for that. Thank you very much.